Hello everyone, this is Daniel Osorio from James Sky Lab of Computational Systems Biology in the Department of Veterinary Integrative Bioscience at Texas A&M University. And today I'm going to talk you about immune profiling at single cell resolution. So in a traditional droplet-based single cell RNA-seq, the experiment starts with the sample collection and preparation, in this case is blood, and then the cells are encapsulated into a drop of oil using the tNE chromium system. The chromium system will take the droplets with all the reagents needed to do the RNA-seq amplification, and they will add each one of the cells in the sample to each one of the droplets, and at the end, the RNA will be amplified and it will send to the sequencer to get the FASTQ files that posteriorly will be used to quantify the expression level of each gene in each one of the cells. So in this four panel of the plot of the figure, we have the expression profile for each one of the cells in each one of the genes. And this matrix can be reduced into 2 dimensions in order to visualize the similarity of the cells and cluster them in order to assign a cell type. So each one of these clusters is representing a cell type based on the coloring. And this is the final goal of the droplet-based single-cell RNA-seq, characterize the cellular composition of the tissues. But if we do that kind of experiments under the right experimental designs, we can get insights about how does the cellular composition of a given tissue change between 2 or more experimental groups. What are the main expression differences in response to a contrasted stimuli? And what is the cell type specificity in the response to that stimulus between 2 different experimental groups? And we can, if we move further to network analysis, we may also able to identify what are the main regulatory differences in response to a contrasted stimuli between 2 experimental groups. But single-cell RNA-seq data, as it is, cannot give us enough information about the adaptive immune system, because single-cell RNA-seq can allow us to characterize the innate immune system consisting in the expression profile of basophils, dendritic cells, eosinophils, mast cells, monocytes, macrophages, neutrophils, natural killer cells, but it may provide us information about the expression profile of the B cells and T cells, but not of all the antibodies and adaptive immune profile that these 2 kind of cells produce. So in response to a pathogen, the B and the T cells are activated, then they start an expansion stage where they divide. Once the pathogen is neutralized, the cells tend to die, and some of them are kept in the system as a memory cells, and those cells are dispersed among all the others. So the ability of the immune system to recognize and bind many epitopes is based on an equal large diversity of antigen receptors. And these antigen receptors is the result of random shuffling and rearrangement of many VDJ segments. So this is constant for both of the cell types, but in B cells also the receptors undergo with somatic hypermutation. In order to be able to quantify this diversity, the tNE company provides a immunoprofiling kit. So what is the goal of this kit? First, being able to quantify the expression of the cells, and additionally to quantify the BCRs and the TCR repertoire in each one of the B and T cells at the same time. It can be done in mouse or it can be done in humans. And today I'm going to explain you how to analyze the data after getting the experiment done. So the first thing that we need is to go to the Tenet Genomics company webpage and to give click in support. Then we give click in single cell immunoprofiling, then in software, and after that we will be able to see the download links. Download links are there and are available for two different things. The first thing that we need is Cell Ranger itself, that is a package to do the alignment of the reads given by the sequencer to the reference genome that in this case we are going to use is enriched for the VDJ segments. So to download the Cell Ranger, we just need to copy this section of the web page and paste it in a bash console of a Linux machine. Then we just need to move to the reference genome that in this case is enriched for the VDJ profiling. And we just need to copy this section and then paste it in a bash console in a Linux machine. These two files are compressed. So the next step is uncompress the files. To uncompress the files, we use the tar and a series of arguments to this package that allow us to uncompress this file. And at the end, we will finish with Cell Ranger /4.0.0 folder. We are going to do the same for the reference. So at the end, we have the two compressed files plus the two uncompressed folders. And for purpose of showing a natural experiment, I'm going to analyze the data set provided by this paper. The link to the data is available following this link in here. And the reported data is available for 2 healthy controls, 5 early recovery stage patients, and 5 late recovery stage patients. But there are 3 more healthy controls reported in the paper. The thing is, the metadata associated with the files is not clear, so we were not able to analyze it in the same fashion. So what we are going to do here is to use the 2 samples that were well reported by the authors of the dataset. So how is the data structured in the web page? For each one of the samples, there is a table like this one. There is a portion of the data that is associated with the expression of the genes. Remember, this is the 5' expression. There is another portion of the data that is associated with the BCR repertoire, and the other is associated with the TCR. So at the end, we will need independently each one of these 8 files. The files are divided in 4 groups because the sequencer has 4 lines. And for each one of the lines, we got a FASTQ file. So at the end, you have the read 1 and the read 2 for the line 1, the read 1 and the read 2 for the line 2, the read 1 and the read 2 for the third line, and the read 1 and the read 2 for the fourth line. The same for the BCR and for the TCR. How to download the data? You click in this blue hyperlink and it will give you an address like this one. So what you can do is to automatize the process of downloading by using this coding structure. So wget is like download, is a package in Linux to download files. You provide the link to the file that you want to be downloaded, and then the -O allows you to save that file using another name. In this case, what we are going to do is to send the downloading file to a folder called FASTQ, and then we are going to add the name of the sample, in this case, the healthy control 2. This is just one sample, it's 1, and it is divided in 4 lines. Remember, line 1, line 2, line 3, and line 4. And for each one of the lines, we have the read 1 and the read 2. The rest is just constant, is 001 fastq.gc. This structure of the name of the file is what allows us to use CellRanger. If the files don't follow this structure in the name, you are not able to use CellRanger because it will identify the files as produced by other company and won't allow you to use it. So CellRanger requires the sample name, the sample one, the line number, the read one and the read two, and the number sequence fastqfile.gc. There is a mistake in here. This should be a 1. And then after we have the CellRanger, the reference, and the data already downloaded in the FASTQ file, we just need to run the package through the command line in Linux. So to do that, we are going to add the binary of the package in CellRanger to the path. So we are able just to type CellRanger and call the package itself. So to export the binary, we just run this line, export path equals CellRanger -4.0.0 path, and then we are able to call CellRanger. CellRanger itself contains a module called VDJ, and VDJ requires these 4 arguments. The first one is the ID. The ID will be the name of the folder created at the end, and it also will be the title of all the reports. The reference will be the folder that we download from the CellRanger webpage. The -F will be the FASTQ files that we already download and submitted to a folder called FASTQ, and a sample that is the name that we use in the files. In this case is AC2, and this match with these three letters at the beginning of the files, AC2. If they don't match, the CellRanger will not be able to identify the files. So this will run, and the time of running will depend on your machine and will also depend on the size of the files. In average, for a medium-sized machine with 60 cores and 200 gigabytes of memory, it will run for 4 hours for this AC2 example. So at the end, you will have a AC2 BCR folder that contains all these files. But the interesting ones, or the ones that we want to analyze, are located in the out subfolder. So if we look at more details in that folder, we have several other files that are intermediate steps in the analysis, the bound files, the FASTQ files, and all the information that is produced during the alignment process, and the A rearrangements that provide all the full sequences of the VDJ rearrangements, the consensus annotations that provide a deep but not as deep as in the A rearrangements. It doesn't provide the full sequences for all the clonotypes, and also a web summary that allow us to identify if the sample was processed in the right way, and how many cells with VDJ rearrangements we were able to identify. So we will start looking at the web summary. The web summary contains the number of cells that we identified, the read of pairs per cell, and the number of VDJ rearrangements that we identify. The important number in here is the reads that map with for any VDJ gene, and also the fraction of the reads that were located in the cells and not out of the cells. So it's not noise in the background. So we have 94% of the reads associated with a cell. And also we have 77 of them associated with a VDJ gene. The consensus file contains all the clonotypes and the clonotypes and all the associated information with it. The B gene, the D gene, the J gene, and also the C gene. We have the amino acid sequence and the nucleotide sequence of the epitope, or of the antibody, the region that is variable, and also the chain, if it's the heavy chain of or the light chain. We have the clonotype. We also have the barcode for each one of the cells, so we were able to trace back which cell contain which heavy chain and which cell contain which light chain. We have that specificity level. And once you have analyzed all the data sets provided by the paper, you may able to start looking at more detail at what are the changes between the differential experimental groups. For example, in this paper, they have healthy control, early stage recovery, and late stage recovery. So to do a more sophisticated analysis over the immune profiling, we are going to use the ImmunoArc package provided by R. And to do that in R, we just need to type install package ImmunoArc. After installing it, we are going to load the package. And once it is loaded, we are going to select the folders. To select the folders, we use a function called list files, and we are going to use a pattern that is -bcr. This because when I process the files, I pay attention to generate all the folders, finishing with that pattern. So it makes my life way more easier afterwards. I just need to tell R to select all the folders finishing in BCR. And as we saw before, we have a OATS file inside each one of the BCR folders. So we need to tell the package where the files are located. So for this case, will be in HC2 BCR OATS folder. So what I'm doing here is to join the name of the folder plus the OATS folder inside each one of them. So this will generate two lists. The first one is the folder list, all the folders with the pattern of BCR, and then a list of files that are the name of the folder plus the OATS subfolder. So that is the direction of where the files are located. Then we are going to move forward and start reading the data. To start reading the data, we can use a for loop, or we can use a lapply function. I sincerely prefer the lapply because it's quicker. So what it needs is a list of files to read, and then a function. This is a variable that we create in order to identify each one of the elements in this list. And then we call the repertoire. This function is included in the ImmunoArc package. And for each one of them, we are getting the data. And from the data, we are getting the consensus annotation. So at the end, what we are going to do is to move in each one of the file lists and guess the consensus annotations for each one of the samples. And we are going to save that in a list called BCR data. In order for this package to work, each one of the list sub list needs to have a name. So we are going to add the names to the BCR data, and they will be the name of the folder. So having that, now we need to add metadata because we want to do analysis about the different experimental groups. So we are going to add the status as a metadata. We are going to copy the folder list into a status list, and then we are going to start replacing. If the folder list starts with HC, healthy control, we are going to replace the name of the folder with healthy. If the name of the folder starts with IRS, so we are going to replace it with early recovery. And the same for LRAs, we are going to say this is a late recovery. So having this list of status will allow us to compare at the end between groups. And at the end, we are going to put all together. We are going to put the data and also the metadata into just one data structure called a list. So we are assembling this in order to all to be together and the package to recognize the final structure of the data. So once we have all the data organized in this way, we are able to start asking the package what are the patterns in the data. So the first thing we can analyze is the clonotype overlap. So to do that, we use the rep overlap function. As input, we use the BCR data and we just provide the data. What this is going to do is to look at the clonotypes that are shared between each one of the samples, and they will color it in function of the overlap values. So how many clonotypes are shared between two of the samples in this matrix. So at the end, you can see that this group of 3 samples share the most number of clonotypes, and all the others are shared in less or more amount, but these 3 are like the most important things or the most stable because they belong to the same group. So I will say the signaling here is very clear. And then you can ask also for the gene that is used during the construction of the different receptors. So the function to use is called gene usage, and then you use the BCR data and the data. But the visualization here is different because we want the visualization to be in function of the status, and the status is inside the meta table that we add to the BCR data. So at the end, you have this plot. When you zoom in, you can see that, for example, for the immunoglobulin K, you have that it is more used in the late recovery stage than in the healthy or the early recovery stage. So you can identify patterns of usage of the gene to generate the different receptors. You can do analysis of the diversity between the samples, and to do that, you may use the rep diversity function. As before, we can plot this in function of the status, and the status is included in the metadata. So this portion of the function is the same, and the only thing that changed with the function in the previous slide is the name of the function. So this plot is produced automatically, and it provides a p-value. In this case, due to the variability of the samples and the low number of samples, just 5 for each one of them, you may not be able to get a statistical significant pattern that you are able to report. So the most important thing in here is being able to detect clonotypes that are present in a group of interest and not in other, and they are expanded, so they are the main basis of the response to the pathogen that the body is responding to. So to do that, we can do the tracking of the clonotypes across samples. And what I'm doing here is to first take all the data, and from the data just selecting the samples, the 1 to the 5 and the 8 to the 12, because these ones are the early recovery stage and the late recovery stage. I'm not interested in the healthy controls. And what I want to get is all the unique amino acid sequences of the receptor that are reported by Cell Ranger. So once I get all the unique sequences, I can do a table to identify which one of them are the most frequent in the table. So I can sort this table from the most frequent to the less frequent, and then select the top 10 more frequent clonotype amino acid sequences and get the names for them. So at the end, my target vector will be a set of clonotypes. In this case, the 10 more frequent in the early recovery stage and late recovery stage between these samples. Then I can use the function track chronotypes. I need to provide what is the data, what are the targets that I want to track, and also what type of columns I want to use. In this case are amino acids. And at the end, I just need to visualize the result of this analysis. So to visualize it, I use the vis function. I provide the input, in this case, the track clonotypes output, and I want the plot to be colorful and smooth. The transitions between the different samples needs to be smooth. So at the end, the final plot looks like this, and this means that these are the 10 more frequent clonotypes between the early recovery stage and late recovery stage. They are less present in the healthy controls. And you can see the proportion of the sample where they are present in each one of them. You can do the same analysis for the TCR, and they will give you other set of clonotypes, because these are the ones expressed by the B cells and the others are expressed by the T cells. And this is all the analysis that you can do using the information provided by the characterization of the BCR and TCR repertoire at single cell resolution. This is the end of our meeting, and I'm going to stop the recording now.